Now we will see what is the biochemical property of apoptotic cell. Means little bit of biochemistry of the apoptosis. So what is the biochemistry of apoptosis? In apoptosis, when we talk about mechanism of apoptosis, if you ask me what is mechanism of apoptosis in a single word, I will describe like this. In apoptosis, there is a one enzyme called as procaspase. Procaspase. In short, I will write procaspase. I will write as a capital C. Right. So this is the procaspase. I'm just writing in short form. Right. So procaspase will be there. This procaspase is inactive form. Right. So what will happen in apoptosis? This procaspase will be activated. Right. This procaspase will be activated to form caspase, right? So I'm writing caspase formation because of activation. So because of activation, it has become caspase. So procaspase to caspase. What is the role of caspase? This caspase will activate. This caspase will activate endonuclease. Endonuclease. And as you can see, the name itself is telling endonuclease will damage the nuclear DNA or chromatin. So because of nuclear DNA damage or chromatin, there will be chromatin condensation. So because of this caspase activating endonuclease, there will be chromatin condensation. Chromatin condensation. And if you remember, just now I have told you, okay, in mechanism of apoptosis, whenever I will write chromatin condensation, means apoptosis is done. So if you ask me, what is the mechanism of apoptosis? In a single word, I will describe caspase activation causing endonuclease activation leading to chromatin condensation. That's all. Caspase, endonuclease, chromatin condensation. This is the single line mechanism of the apoptosis, which we will see and we will elaborate here onwards. But before that, I want to tell you important point regarding the endonuclease. Another important MCQ in exam. Endonuclease. As I have told you, ki endonuclease, their basic target is nuclear DNA damage, right? So this nuclear DNA damage is random, or they will cause a specific fragment removal. That is important. Is it random or a specific fragment? So your answer will be ki nuclear DNA damage by endonuclease will be a specific fragment will be removed. So here you will observe that they will be removing a specific fragment. Specific fragments, right? Specific fragments of nuclear DNA will be removed, and these specific fragments are known as internucleosomal DNA damage. So that was all India question. Internucleosomal DNA damage. So internucleosomal DNA damage is feature of apoptosis. That was the question in older All India exam and AIIMS exam. This was the favorite for examiner. Internucleosomal DNA damage is a feature of apoptosis. So just keep that in mind. Internucleosomal DNA damage is a feature of apoptosis. So this is the older question. What will be the future question? If you read Robbins, you will find internucleosomal DNA damage. They will be consisting of 180 to 200 base pairs of the DNA. That will be the future question in NEET exam, right? So examiner may ask question like this: internucleosomal DNA damage. What is the fragment size which has been removed? So internucleosomal damage, size of fragment will be multiples of 180 to 200 base pairs, right? So they will be always into multiples of, see what I'm writing, into multiples of 180 to 200 base pair, and that is how question will be asked in future exam. So internucleosomal DNA damage is not a random damage, it's a specific damage, a specific cleavage of the DNA that will be multiples of 180 to 200 base pairs, right? So when this base pair will be removed, we can observe them on agarose gel electrophoresis, right? So this we can observe them on a special method which is called as agarose gel electrophoresis. Agarose gel electrophoresis. electrophoresis. On agarose gel electrophoresis, what we are going to observe, I will just show you one diagram of agarose gel electrophoresis. Suppose I am showing you this agarose gel electrophoresis. This is the agarose gel electrophoresis. I will show you this diagram, just a schematic diagram, which I wanted to demonstrate here. In agarose gel electrophoresis, there are three possibilities we will observe, right? Number one, because we are talking about the normal cell also. So normally you will see a simple cell like this, which will be opaque area. So this is the normal cell, right? Second possibility is you will see DNA damage by endonuclease in a fragment which is getting smaller and smaller like a ladder. Stepwise getting smaller like a ladder. Stepwise getting smaller like a ladder. So this pattern is called as a step ladder pattern. So remember, this is called as step ladder pattern. So on agarose gel electrophoresis, we are seeing a step ladder pattern. A step ladder pattern is caused by endonuclease. So remember, who is causing this step ladder pattern? We have to remember the reason for step ladder pattern is endonuclease, right? Because endonuclease is causing damage of the DNA into fragments of 200 base pair, 200, 400, 600. So that is why they will be in a step ladder pattern, 200, then 400, then 600, right? 200, 400, 600, 800. Like that it is increasing. That is why it is called as a step ladder pattern. A step ladder pattern is a very important MCQ in older AIIMS and All India exam. See, this language is very important. When you are studying any topic in your textbooks, we must focus on the language. What language examiner may ask from those areas? See, I am going to demonstrate you one example. A step ladder pattern, when examiner will ask you this language, a step ladder pattern is seen in. So what should be your answer? A step ladder pattern is seen in apoptosis or necrosis. See this, examiner is asking a step ladder pattern is seen in. Step ladder pattern is seen in option number 1 is apoptosis, option number 2 is necrosis, and option number 3 is both of the above. So what should be right answer? See the language, seen in. When examiner is saying seen in, it will be seen in both of them, right? A step ladder pattern is seen in both apoptosis and necrosis, but when language is step ladder pattern is characteristic of, then your answer will be only apoptosis. So that is the language we have to be very, very watchful during exam time. And this language had been used in AIIMS exam, right? Step ladder pattern is characteristic of apoptosis. Because of which enzyme? Endonuclease. And that was the answer, because of endonuclease. So a step ladder pattern is characteristic of apoptosis because of the presence of endonuclease. Step ladder pattern is seen in both apoptosis and necrosis for future All India exam. So this image you can see, I have shown you a step ladder pattern, but there is another possibility where you will see this plate will be having feature like a smear. See this is like a smear, right? This is called smearing pattern. This pattern is called as smearing pattern. AIIMS May 2016. This was the question. Smearing pattern is seen in which of the following conditions? So this was the AIIMS question. So smearing pattern is seen in necrosis, and that was the answer in AIIMS exam, right? So smearing pattern is seen in necrosis. This was the answer. Smearing is seen in necrosis. So now we can see that agarose gel electrophoresis, there are three things: normal, step ladder, and smearing pattern. So smearing pattern was AIIMS question in 2016. Now I'll demonstrate the diagram. You can see this picture of the agarose gel electrophoresis, as I have discussed. Okay, when examiner will ask you this question, so you can see this is the number 1, and examiner will ask you what is number 1. It's normal. What is number 2? This is how question will be asked. This number 2 demonstrate a step ladder pattern, right? We can see there is a step ladder pattern. You can see step ladder pattern. So step ladder pattern is characteristic of apoptosis, but remember, not diagnostic. Characteristic but not diagnostic. Why it is not diagnostic? Because it will be seen in necrosis also. And the third pattern, which was the question in AIIMS exam, this is the diffuse smearing pattern which we will see in necrosis. So this is the agarose gel electrophoresis in an apoptosis, right? So we have seen the biochemistry of the apoptosis is revolving around mechanism of the apoptosis, where we are seeing caspase is getting activated. They are activating endonuclease, and endonuclease is causing nuclear DNA damage, causing chromatin condensation, and that is how apoptosis will happen. So in simple words, caspase activation, endonuclease activation, and chromatin condensation is the mechanism of apoptosis, right? So now we are going to discuss in detail about mechanism of apoptosis. So we are going to discuss detail about mechanism of apoptosis. What is the mechanism of apoptosis, right? So in mechanism of apoptosis, already I have told you caspase, endonuclease, chromatin condensation, but now I am going to bifurcate them into two events: initiation and execution. So there are two events in mechanism of apoptosis. So there are two events. One is called initiation and second event is execution. So both initiation and execution, they will be involved with caspase activation. Different caspase will be activated in these pathways. So let us see what caspase will be activated. To understand that, again I will tell you that initiation event will be consisting of two pathways, right? One pathway is called as intrinsic pathway. See how I had made I. This is a mnemonic. I looks like 9, right? So this is the intrinsic pathway. Intrinsic. So as I said, there are two pathways. So I'm just showing you these are the two pathways, right? Intrinsic pathway and then extrinsic pathway. Intrinsic and extrinsic. So these are the two pathways which we are going to see in the initiation event, right? Intrinsic and extrinsic pathway. Intrinsic pathway, caspase 9 will be activated. See this? I looks like 9. So intrinsic pathway, caspase 9 will be activated. Extrinsic pathway E for 8. So caspase 8 will be activated in extrinsic pathway. But here is a twist, which was the question in All India 2015. In human being, extrinsic pathway, which caspase is activated? Because caspase 8 is seen in lower animals, not in human being. They are present in lower animals. They are not present in human being. So in human being, which one is seen? So remember, in human being, caspase 10. So caspase 10 is for human being, right? So in humans, caspase 10 is the main caspase in the extrinsic pathway, right? So in intrinsic pathway, caspase 9, extrinsic pathway, caspase 8 in lower animal, caspase 10 for human being. That was the question in All India exam, right? So now we can see during initiation, caspase 9 and caspase 8 and 10 is activated from the procaspase. So procaspase 9, procaspase 8, procaspase 10 has been activated to form active caspases. That is the initiation. So after initiation, what will happen? After initiation, all these caspases will be activating executioner caspases. See this, what I said? All these caspases, C9, 8, 10, these are the initiation, right? So initiator caspase will be activating executioner caspases. What are the executioner caspases? These are the C7, C6, right? So 6 and 7, these are two executioner caspases. Apart from this, one more name you have to remember, C3. So 3, 6 and 7, these are the three executioner caspases which will be activated in executioner pathway. So once they are activated, when executioner caspases are activated, now all together they will be activating endonuclease, and because of the endonuclease activation, nuclear DNA will be damaged and there will be chromatin condensation. There will be chromatin condensation, which I said is most characteristic equivalent to apoptosis, right? So that is the mechanism of apoptosis: caspase, endonuclease, chromatin condensation, which I have divided into two events: initiation, execution. Initiation is further subdivided into intrinsic, extrinsic pathways. Intrinsic pathway caspase 9, extrinsic pathway caspase 8, right? And now we can see all these initiation caspase, they are activating executioner caspase, and now they are activating endonuclease, causing chromatin condensation, and that is the apoptosis, right? In simple way. Now again, we will move to the detailed discussion about these two pathways, intrinsic and extrinsic pathway. But before that, I would like to give you one concept regarding the proto-oncogene. In our human body, remember this point, this concept of the proto-oncogene is very important to understand apoptosis. Proto-oncogene. Proto-oncogene, as we all know that these are the normal gene present in our human body. Proto-oncogene is a normal gene present in our human body for cell proliferation and differentiation. So proto-oncogene is a normal gene required for cell proliferation and differentiation, and proto-oncogenes can be of two categories. This can be pro-apoptotic. Pro-apoptotic. Pro-apoptotic means who will promote apoptosis. That is how we can understand. Pro-apoptotic means who will promote apoptosis. And second group of proto-oncogenes are anti-apoptotic. Anti means which will prevent, anti-apoptotic genes, right? So now we can see there are two group of proto-oncogenes present in our normal human body. One which will promote, called as pro-apoptotic. Second, which will prevent, they are called as anti-apoptotic genes, right? So we will see what are the pro-apoptotic and anti-apoptotic. Pro-apoptotic genes are BAX, right? So these are the pro-apoptotic genes. What are the anti-apoptotic genes? In anti-apoptotic gene, one important name which examiner had asked in this All India exam was BCL2, one of the very frequently asked question in exam. BCL2 is an anti-apoptotic gene, prevents apoptosis, right? Another anti-apoptotic gene is the BCLX. But here there is a twist, and that twist is there are two types of BCL, right? BCLX, right? Who is pro-apoptotic, who is anti-apoptotic? So that is important point to focus upon, because examiner will ask question like this only. BCXL will be an anti-apoptotic, BCLXS will be apoptotic, pro-apoptotic, right? S means just mnemonic I am telling you. S which will stimulate the apoptosis, S stimulates. L which will lower the apoptosis, lowers. So this is how we can remember this by mnemonic, right? L means they will lower the apoptosis, S means which will be stimulating the apoptosis. So BCLXS is the stimulator, BCLXL is the anti-apoptotic. This is how we can understand, we can remember this point, right? Apart from this, there is one more anti-apoptotic which examiner may repeat or may ask in future exam, that is the MCL1 gene. So MCL1 is another anti-apoptotic gene which we have to keep in our mind. So there are three important anti-apoptotic genes: BCL2, BCLXL and MCL1, right? Pro-apoptotic also we are remembering three: BAX, BCLXS. Apart from this, there is a fourth member here which we have to remember, which is called as BH3 only proteins. BH3 only proteins family. BH3 only proteins family. So this is the another important pro-apoptotic protein. What is the BH3 protein? What is the BH3 only protein family? This is the family. This is a group of proto-oncogenes, right? In this family, right? So what are these BH3 only proteins family? These proteins are, or these proto-oncogenes members are, as I said, there are 5 members. which we have to remember like this. There is a mnemonic, a lady, her name is Bimla. So I'm just writing Bim because number 1 is Bim. So I'm giving a mnemonic. Bimla is bad in bidding. So see the mnemonic what I'm writing. Bimla is bad in bidding. Bim, bad, bid, right? Bimla is bad in bidding whenever she goes to the share market. Why she is bad in bidding? Because she often goes for one company, right? So that is the Puma, and then is another company is Noxa, right? So Puma and Noxa. So Bimla is bad in bidding for two companies. That is why she often bear the losses, right? She don't know when to bid, when the proper time will come, and that is why she is poor. So Bimla is bad in bidding, Puma, Noxa. So these are the 5 BS3 only protein families, right? And BS3 only protein family, they are present on the cell surface. So that is the another important point we have to keep in mind. They are present on the cell surface. So BS3 only protein present on the cell surface. Members are Bim, Bad, Bid, Puma and Noxa, right? So whenever Bimla goes for the share market bidding and she is bad, so her husband gets stress, right? So that is why these are stress sensors of the cell. So whenever cell is under stress, this will be induced. That is why examiner can ask one important future MCQ. Who are the stress sensors of the cell? Who are the stress sensors of the cell? Could be a future potential MCQ in exam, right? So stress sensors are BS3 only proteins which are present on the cell surface. So that is how we can remember stress sensors are the BS3 only protein which is present on the cell surface. So this concept of proto-oncogene is very, very important to understand, because earlier in All India exam there was a question कि proto-oncogenes can induce apoptosis. Answer is yes. But suppose examiner twist the language कि proto-oncogenes will prevent apoptosis. Answer will be again yes. That is why, see, that is why I wanted to classify like this कि proto-oncogenes are both apoptotic and anti-apoptotic. So always keep that in mind कि proto-oncogenes are both apoptotic and anti-apoptotic, right? So now we will move ahead and now we will discuss detail about the initiation, intrinsic pathway and extrinsic pathway. So now we are going to discuss in detail about intrinsic pathway and extrinsic pathway. So as I said, these are the part of initiation. See, we started like this, right? There is a one event called initiation, and already we have discussed, right? Initiation is having two pathways. So number 1 we are going to discuss, that is intrinsic pathway. So number 1 is intrinsic pathway. So we will start discussing from the intrinsic pathway, right? So in intrinsic pathway, what is another name of intrinsic pathway? Intrinsic pathway is also called as mitochondrial pathway. Intrinsic pathway is also called as mitochondrial pathway because mitochondria is the most important organelle for this pathway, right? So this point we have to keep in mind. This was the All India question also, right? कि mitochondria is the most important organelle. Mitochondria is the most important organelle for intrinsic pathway, right? And intrinsic pathway is the major pathway also. So sometimes this is also called as major pathway. So these are the two important MCQ which examiner had asked earlier in All India exam. Most important and second is the major pathway is the intrinsic pathway, right? So intrinsic pathway, to start intrinsic pathway, first I would like to tell you कि why a normal cell is not having a spontaneous apoptosis. A normal cell, see what I said, a normal cell. So just imagine I am drawing a normal cell. This is the normal cell and this is the mitochondria. Mitochondria, outer membrane, and now I am drawing inner membrane. Inner membrane will be having invagination which is called as cristae. See, there are two cristae I have drawn for a purpose. So you also draw two cristae, right? So we were talking about a normal cell, why there is no spontaneous apoptosis. So answer is because outer membrane of the mitochondria, outer membrane, see outer membrane of the mitochondria have drawn, and here I am saying that there is an anti-apoptotic protein called BCL2 or MCL1 which prevents apoptosis, right? So remember, outer membrane of the mitochondria will be having anti-apoptotic proto-oncogene that will not allow spontaneous apoptosis. That is the number one reason. And inner membrane usually have the apoptotic genes like BAX or BAC. So that is hidden. So apoptotic are on inner membrane, anti-apoptotic on outer membrane. That is why there is no spontaneous apoptosis in a normal cell. Apart from this mitochondrial activity, cytoplasm is also involved. Cytoplasm is also involved. Cytoplasm is having another important cytoplasmic protein which is called as inhibitor of apoptosis, IAP, inhibitor of apoptosis. So this is the another protein which is present in the cytoplasm. So this also prevents apoptosis. So that is why normal cell is not having any spontaneous apoptosis, because of the outer membrane anti-apoptotic gene, and cytoplasm is having another important protein known as inhibitor of apoptosis. So whenever cell is under stress or gets the injurious stimuli, they will undergo apoptosis. So whenever cell is having a stress or stimuli, that stress or stimuli can be anything, right? It can be, as I said, this stress or injurious stimuli can be anything, right? Stress or injurious stimuli, right? Injurious stimuli, we know it may be radiation injury, it may be physical injury, it may be chemical injury. Whatever kind of stress we are providing to a cell, what will happen? This stress will activate. There is a protein present on the cell surface. What is name of this protein which is present on the cell surface? You remember? Whenever stress will be there, you remember stress sensors are present on the cell surface. So what is the name of this stress sensor we are seeing here is the BH3 only protein. So BH3 only protein is a stress sensors present on the cell surface. So stress will be activating this stress sensor. See what I am saying? Stress will be activating this stress sensor which is BH3 only protein, right? And once BH3 protein has been activated, they will be having two functions. Dual function. So what are those dual function? Number one, BH3 protein will inactivate the BCL2. So they will inhibit BCL2 on outer membrane of the mitochondria. And second, they will activate the BAX and BAC. So they will activate the apoptotic gene which is present on inner membrane. Because of the activation of the BAX and BAC, now they will be expressed on outer membrane. Right? So BAX and BAC is getting expressed on outer membrane. Because of the BAX and BAC expression on the outer membrane, what will happen? Because of the expression of the BAX and BAC, now this mitochondrial membrane will become permeable. Mitochondrial membrane will become permeable. So permeability of the mitochondria is increased, right? So because of that, there will be increase in mitochondrial permeability. Increased in mitochondrial permeability is a feature of apoptosis. This was the PGI question in 2012, right? So mitochondrial permeability increase will be seen in apoptosis. So because of the mitochondrial permeability has been increased, so what will happen? There are two important events. Number one, mitochondria, they are having cytochrome C. This is the cytochrome C. Cytochrome C normally resides in the mitochondria and their role is in electron transport chain. So when nothing is mentioned in exam and examiner says cytochrome C function is for electron transport chain. So I am writing here, when examiner will be giving you question like this, what is the cytochrome C function? You will say that it is for electron transport chain oxidative phosphorylation. So when nothing is mentioned here, it should be considered as mitochondrial cytochrome, right? That is what I wanted to tell you, because this is a great language which examiner had used in AIIMS and All India both exam many times. Cytochrome C main function is electron transport chain, but in case of apoptosis, what we are observing, because of the mitochondrial permeability channel has been formed, so this cytochrome C will leak outside, and now in the cytoplasm they will be forming cytosolic cytochrome C, and that was the question in All India exam. So now you can see the language, what language examiner is using. Cytosolic cytochrome C, what is the function? And that was the question, cytosolic. So see the language, cytosolic, not mitochondrial. Mitochondrial is electron transport chain, but when they are talking about cytosolic cytochrome C, it will induce apoptosis. That was the answer. How? Another question in All India exam, right? So when cytosolic cytochrome C will combine with which protein? Apoptosis activating factor 1, right? So apoptosis activating factor 1. So that is the important thing we have to understand, right? So cytochrome C, which has leaked out into the cytoplasm, now they will combine with APOP1 protein, and this combination is known as, that is the another important MCQ which examiner asks, right? This is known as cytosolic cytochrome C, and APOP1 is called as apoptosome. This is called as apoptosome, right? So this is known as apoptosome. What is the function of apoptosome? That is another important question. Function of apoptosome is to cleave procaspase, or you can say it has to activate. So apoptosome will activate procaspase. Which procaspase? My question is, which procaspase? Which procaspase? We are talking about intrinsic or extrinsic pathway? Intrinsic. Looks like 9, right? So procaspase 9 will be activated to form caspase 9. So this is how caspase 9 activation will be done by apoptosome. But remember, this is not that easy, because in the cytoplasm there is another anti-apoptotic protein called as IAP. So IAP has a tendency to inhibit at this step. So remember, this IAP has a tendency to inhibit this apoptosome function. So they will inhibit apoptosome function, right? So that is what the main function is of this IAP, to inhibit apoptosome functions so that they cannot activate procaspase 9 into active form of caspase 9, right? So how this apoptosis will start in intrinsic pathway? So how this mean to say that how this intrinsic pathway will be completed, because this is blocking it. So to activate or to finish off this intrinsic pathway, come back to the another permeable channel in mitochondria. So through this permeable channel of the mitochondria, now there will be another enzyme. Now there will be another enzyme from the crusty of the mitochondria which will leak outside, and this will block this IAP. So what is this enzyme? This is called as SMAC diablo. SMAC diablo protein. So this SMAC diablo protein, which is present in the mitochondria, will come outside, right? And now they will inhibit this IAP. So this was the question in JIPMER exam also, and previous year All India exam also. What is SMAC diablo? Question was, what is SMAC diablo? You tell me. What is SMAC diablo? Will you consider them as a pro-apoptotic or anti-apoptotic? Now look at the concept. What we are seeing in this concept, in this diagram, concepts are very clearly demarcated. SMAC diablo was present inside the mitochondria, came outside, it has blocked the IAP. IAP is the anti-apoptotic, right? And when anti-apoptotic has been blocked, so what will happen? This apoptosome will convert activation of the caspase 9. So SMAC diablo is pro-apoptotic, and that was the answer. So that was the answer. They are pro-apoptotic compound or enzymes, right? So SMAC diablo is pro-apoptotic enzyme released from the mitochondria, right? So this is how you can see the intrinsic pathway in a simple manner. I will again repeat, right? Because of the stress sensors were activated, which were BS3. Stress sensor BS3 has inactivated BCL2 and activated BAX-BAC. Because of the activation of the BAX-BAC, we had observed there is a permeability channel formed in the mitochondrial membrane, which will allow the leakage of the two important proteins, cytochrome C and SMAC diablo. SMAC diablo and cytochrome C. Cytochrome C, when they will come outside, cytochrome, that will be called as cytosolic, which will combine with APOP1. and this APAF1 will be combining with cytochrome C, and they will be forming a substance called as apoptosome, which has tendency to activate caspase 9, but this will not happen because of the IAP. So IAP will be inhibited by SMAC-Daplo, another protein in the mitochondria. So SMAC-Daplo is also pro-apoptotic protein. So like this, now we can see in the end, caspase 9 has been activated by the intrinsic pathway. So this is the intrinsic pathway, right? Now we will move to the next pathway, which is extrinsic pathway, right? So second pathway we are going to discuss is the extrinsic pathway of apoptosis. As I have told you, intrinsic pathway was the major pathway, mitochondrial pathway. Same way, extrinsic pathway is also called as death receptor mediated pathway. So they are called as death receptor mediated pathway. So extrinsic pathway is death receptor mediated pathway. What are these death receptors? Now the question is, what are these death receptors? So there are numerous death receptors, but I will enumerate two important death receptors in our human body. Two important death receptors are, number one is the tumor necrosis factor receptor, right? Tumor necrosis factor receptor, number one death receptor, and second death receptor is the FAS. So these are the two important death receptor. There was a question in older All India exam, which is the best defined death receptor in human body, or most well defined. So best defined death receptor, tumor necrosis factor receptor, is the best defined death receptor in human body. That was the question, right? So that was the question in one's All India exam. Best defined is the tumor necrosis factor receptor, right? So this is the best defined. FAS, what is another name of FAS? Another All India question which was asked in 2014 and 15. FAS is also known as, remember, FAS is also known as CD95. FAS is also known as CD95, or you can say that CD95 is a marker of FAS death receptor. What I said? CD95 is a marker of FAS death receptor, because this was the exact question in All India exam. Which is the marker of death receptor? Which of the following is the CD marker of death receptor? So CD95 is a marker of death receptor, and that death receptor is the FAS, right? And this was the old question also in All India exam, AIIMS exam. Which CD marker is marker of apoptosis? Which CD marker is marker of apoptosis? So CD95 is a marker of apoptosis, extrinsic pathway, right? What happens in extrinsic pathway? As I have told you, these are death receptor mediated pathway, and suppose I am showing you a cell. This is the plasma membrane, right? This is the lipid bilayer plasma membrane. So on plasma membrane, there will be location of the death receptor. So here I'm just showing you, this is the death receptors, right? So these are the death receptors. In the cytoplasmic border, these death receptors are attached to the another protein. It is attached to the another protein which is called as adapter protein, right? Adapter protein. And this adapter protein is attached to the procaspase. Which procaspase? Extrinsic pathway 8, or in human being 10. So this is one basic architecture of the cell. Death receptor is attached to adapter protein in the cytoplasm, which is holding procaspase 8 in inactive form. So you can see it is inactive because it is pro form, right? So what happens whenever death ligand will enter? Whenever death ligand will come and they will combine with their respective death receptor. For example, it can be anything, right? So just take one example. If it is a tumor necrosis factor receptor, which will combine with tumor necrosis factor receptor ligands, right? So this is the complementary ligand. So whenever this death receptor combines with death ligand, this will activate the adapter protein, right? So because of this will be activated, right? So this will activate the adapter protein, and this will activate the procaspase 8. and this will be converted into active form of caspase like this, right? So whenever complementary ligands combine with death receptor, adapter protein in the cytoplasmic membrane will be activated. This will convert procaspase into active form of the caspase, and now you can see that C8 and 10 has been formed. So this is the simple. So death receptor pathway or extrinsic pathway of the apoptosis is a very simple pathway, as we can see here also, right? But here I can tell you one thing. In a normal human being cells, there is a one protein which was the question in JIPMER exam previous year in All India exam. Which protein is anti-apoptotic? Which protein present in our normal human cell is anti-apoptotic? So here I want to tell you that important name of the protein. That protein is called as FLIP. So FLIP is one protein. FLIP is a protein present in normal human cells as well as certain viruses like human immunodeficiency HIV viruses. So our normal cells and certain viruses like HIV viruses, they have one unique protein which is called as FLIP, and FLIP is an anti-apoptotic. So that was the question in All India exam and JIPMER exam. What is FLIP? FLIP is an anti-apoptotic protein, right? So FLIP is an anti-apoptotic protein. That was the right answer. So now the question is, what will be the future question? How examiner will be asking question in future exam? FLIP is an anti-apoptotic protein which will inhibit apoptosis in which pathway? Intrinsic or extrinsic? So now we are seeing extrinsic pathway. So FLIP is for extrinsic pathway, number one. And number second, future question will be at what step they will inhibit? They will inhibit adapter protein activation or they will inhibit procaspase to caspase activation. That is how question will be asked in exam, and that's the point we have to keep in mind. FLIP will inhibit activation of the caspase. So FLIP inhibits activation of the caspase in extrinsic pathway. That is how we can anticipate a future MCQ. This is how future question will be asked. FLIP will be inhibiting extrinsic pathway, activation of the caspase, right? So now we can see that this entire extrinsic pathway is activated. So initiation is completed, right? So intrinsic pathway, extrinsic pathway, both are activated. So initiation has been completed, right? Initiation has been completed. So again, I will sum it up. So initiation has been completed because intrinsic pathway, because extrinsic pathway, both are activated, and that is why intrinsic pathway C9, extrinsic pathway C8 and 10 has been activated, right? So because of the initiation, because these two things are activated, now they are going to activate executioner event. So execution. So in execution, which caspase we will see? Caspase 3, 6 and 7. So this is how execution will be started. So initial C9, C10, 8 will activate executioner caspase, and these executioner caspase will activate endonuclease, and after that we all know that there will be chromatin condensation, which is equivalent to apoptosis, most characteristic feature. So that is how I begin. Caspase, endonuclease, chromatin condensation is the single line mechanism for the apoptosis, which we have elaborated and we have seen the mechanism, right? Now here I want to tell you few important MCQs which examiner ask in exam very frequently, where you have to be very careful. For example, what is hallmark of apoptosis activation? So what is hallmark of apoptosis activation? One of the very frequently asked question in All India and AIIMS exam. Which enzyme activation, endonuclease or caspase? So answer is caspase activation. So whenever you find caspase activation is hallmark of apoptosis activation, right? So whenever procaspase is getting converted into caspase, that will be considered as apoptosis hallmark, right? So now the question comes to our mind. There was a DNB question in 2014. It's a beautiful question, and one should be very careful for the language. That language was, What is the hallmark of neuronal apoptosis activation? Look at the language. Look at the language which I was talking about. What is the hallmark of neuronal apoptosis activation? So what is the hallmark of neuronal apoptosis activation? So this was the question. And here you can see the language has been changed. It's not the simple language. Here examiner has introduced a new word that is called neuronal. Because in neurons there are no caspases. That's all. Right? Neurons there are no caspase. So caspase activation is not for the neuronal apoptosis activation. In neurons there are different type of caspases, and they are called as different type of substances, which is known as apoptosis inducing factor, which is known as AIF activation. So hallmark of neuronal activation will be AIF activation, where AIF stands for apoptosis inducing factor. So apoptosis inducing factor activation is hallmark of neuronal apoptosis. So that is the important thing we have to remember. Okay, when examiner says a simple language, hallmark of apoptosis activation, caspase activation. But when examiner says what is hallmark of neuronal apoptosis activation, your answer should be apoptosis inducing factor activation. Right? What is the clinicopathological correlation of this apoptosis to the neurodegenerative disorders or some other disorders? Right? Here I would like to tell you one important thing. Whenever, in simple words, I will just tell you what is the clinicopathological correlation, right? So this is one important information I wanted to tell you, because we study about apoptosis, but we don't know how to apply this knowledge to our clinical knowledge, right? How to evolve our clinical knowledge on the basis of these simple general pathology facts and figures, right? So here I would like to tell you, whenever there is a misfolding of the protein going on, right? Misfolding of proteins are going on. So this misfolding of proteins, what they are going to do, they are going to activate caspase, right? They are going to activate caspase. This caspase will induce apoptosis in those concerned tissues, right? Or in those concerned cells. But when this caspase is not induced, suppose this caspase is not induced, right? When this caspase is not induced because of the misfolding of the protein, caspase is not induced. So what will happen? There will be accumulation of misfolding protein, or you can say misfolded protein will be accumulated. So this is the important thing. Whenever misfolding is more in our human body, this will activate caspase in normal condition, and this caspase will induce apoptosis. So see this, what I have shown you here, this is the normal condition, right? What I am showing you here, this is the pathological condition. Misfolding protein has been accumulated, and this is feature of many of the neurodegenerative disorders. So see what I said, this is the feature of neurodegenerative disorders. For example, if examiner asks you Alzheimer's disease, is it a misfolding protein disorder? Answer is yes. This was the All India question. Alzheimer's disease. Parkinson's disease, answer is yes. Parkinson's disease is another example of the misfolding of the protein, right? Huntington's chorea, right? So these are the three important neurodegenerative disorders where misfolding of the protein is the basic pathology. See what I have written? Misfolding of the protein is the basic pathology. Alzheimer's disease, Parkinson's disease and Huntington's disease, they will be having increased accumulation of the misfolded protein. So that is the relevance of the apoptosis. When apoptosis is defective, we may see this kind of neurodegenerative disorder due to accumulation of misfolded proteins, right?